I think there's plenty of reason why to explore them in early on. I think for our especially high-risk myeloma, we don't do well. Perhaps that patient can be called out for a new treatment paradigm. Also, your immune system is probably more conducive to producing these effectively when you haven't had too much treatment. And one thing fortunate is that in a lot of the trials to date with CAR T cells, we haven't seen that CRS and neurological toxicity that has plagued some of the other developments. Not that we are totally free of that with the BCMA products. Some are more, toxic than others in the experience to date. However, I think the toxicity profile that has emerged justifies that it could be moved into earlier lines of treatment.
Yeah, I think that, at least in my own personal experiences, and while I'm not a lymphoma doctor, I get to cover the lymphoma patients who are going through CAR-19 therapies. The level of toxicity, the complications are clearly much less with the BCMA-directed CAR T cells, at least what we're using in our local institution. So I think we have every right to be excited about this. I think we should be even more excited that this is going to drive additional target selection, whether one of the targets that is already being investigated is going to turn out to be the best one, whether it's going to be SLAM F7 or CD56 or CD38, whatever the target might be. It's going to be fun. I mean, at least from the sense that we're going to have an opportunity to control disease in patients that right now we're just throwing up our hands and not really having great expectations.
No, I agree. I think there is a lot unknown. Not only the targets, we don't know what the right dose is, how should you give more than one product, whether persistence is needed, what are the right co-stimulatory molecules. In the future, we will probably have CARs perhaps directed at more than one antigen, and so I think that there is potential for this technology going forth. We haven't obviously, I think, maximized its potential in any way.
Yeah, and I think that there will be an economy of scale, so hopefully this will become more accessible to more people.
I think it's likely as well that there'll be some level of enthusiasm to combine these agents with other immune drugs. And I think we need to be very cautious in that space because we don't want to have a catastrophic outcome that could impede the development. So I think there needs to be very careful thought about what to combine with, and for there to be a rationale rather than just, we think this is a good idea. I think we need to be very cautious because we don't want to, push things off the rails in terms of development.
Yeah, I think one of the things that strikes me the most about this, and this is not necessarily specific to CAR T cells, but that you and I are perhaps slightly older than Ravi, but
not by much.
But it wasn't that long ago that the idea that one company would invest this kind of dollars and infrastructure in developing this kind of technology in myeloma, let alone to have 10 or 15 or 20 companies out there competing for this space, spending this kind of money on myeloma. As I said, it wasn't that long ago when no one was interested in myeloma as a target disease because there wasn't enough financial motivation to do it, and now we're lucky. I mean, that we're having this kind of conversation about this kind of technology in our disease.
It raises an interesting point because I think a lot of the drug development in myeloma is because of the remitting, relapsing nature of the disease.
Yeah.
In that, there's money in their hills with a drug that'll work in relapse disease. And the paradigm here might be completely different. So it may actually make all of that development redundant if the right target and population are identified.
Yeah.
I think one thing that could make it feasible in the future is if you can have an off-the-shelf product that you can bring the cost down with. That's another area where I think the future may be.
Yeah. And it may not be CAR T cells that we end up with. It may be, CAR NK cells. It may be, we're just beginning to scratch the surface. I don't think any of us would argue that it's a very exciting time for us, but more importantly for the patients.
Very true.
So I'd like to thank Ravi and Andrew for joining us. I think that this is an extraordinarily important subject in myeloma, but in cancer biology in general, and I appreciate the opportunity to talk about this. So thank you guys very much.